Greetings! This is my first video of the Everything You Don't Need to Know series. Let's begin with something basic and make it complicated: the cell cycle. Mitosis. The cell cycle is a succession of events that lead, in its shortest definition, to make two cells out of one. But this is not all. The cell cycle is a process that's led by both external and internal regulators, and it is far more complex than one cell makes two statement I made earlier. So you can get the general idea. In eukaryotes, cells that have a nucleus can form multicellular organisms like animals. This cell cycle I'm referring to is based on humans. The cycle is often depicted as a circular process. This is due to the cell cycle control system, where each cell stage of the cycle has an order, a process that happens inside it, and its regulators. This is regulated by cycle-independent kinases, CDKs, that join proteins called cyclins. It's circular because it repeats over and over, except in certain circumstances where the cells become arrested. As an outline, cells spend most of their time in interface, which is divided into G1, S, G2. Both G's are growth and S is for synthesis. There is also mitosis, or M, which is subdivided into prophase, metaphase, anaphase, and telophase. Not always in the cell cycle you have the G's. For example, in early embryological development, the divisions are so fast the cycle skips over the G's and goes straight to S and mitosis repeatedly. Also, in cell populations like nerve and muscle cells, there's an additional stage called G0 where cells arrest. Each cell cycle phase also has checkpoints. In interphase is where cells spend most of their time. Let's go to the details now. G1 is where most cells become committed to either continued division or exit from the cell cycle. In unfavorable growth conditions or with inhibitory signals from other cells, cells may pause for extended periods in G1 or even enter G0 or the arrested state. In G1, in this phase, the cell accumulates the building blocks of chromosomal DNA and the assorted proteins, as well as accumulating sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. In S is where DNA duplication happens and two sister chromatids appear. It's also where usually centrosomes duplicate. Centrosomes are associated with centrioles, which are involved in cell division. G2. In this phase, the cell replenishes its energy stores and synthesizes proteins necessary for chromosome manipulation and movement. It's also where DNA repair events happen. Some cell organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the mitotic phase. Mitosis is where cells divide. The first portion of the mitotic phase is karyokinesis, or nuclear division. The second portion of the mitotic phase is called cytokinesis, which is the physical separation of the cytoplasmic components into the two daughter cells, which end up with 46 chromosomes. In G1, chromosomes are 46. There are 46 chromosomes in U1. In S, they duplicate into 92. And then after mitosis, it goes back to 46 chromosomes. Phases of karyokinesis are prophase, prometaphase, metaphase, anaphase, and telophase. Prophase, or also called the first phase, is where the membranous organelles, such as the Golgi complex and the endoplasmic reticulum, fragment and disperse towards the periphery of the cell. The nucleolus disappears as well, and the centrosomes begin to move to opposite poles of the cell. Microtubules that will form the mitotic spindle extend between the centrosomes, pushing them further apart as the microtubule fibers lengthen. The sister chromatids begin to coil more tightly with the aid of condensate proteins and now become visible under a light microscope. During late prophase, the nuclear envelope begins degrading. Prometaphase, the first change phase. Many processes that began in prophase continue to advance. The remnants of the nuclear envelope fragment further, and the mitotic spindle continues to develop as more microtubules assemble and stretch across the length of the former nuclear area. Chromosomes become even more condensed. Each sister chromatid develops a protein structure called a kinetochore in its centromeric region. The proteins of the kinetochore attract and bind to the mitotic spindle microtubules. As the spindle microtubules extend from the centrosomes, some of these microtubules come into contact with and firmly bind to the kinetochores. Once a mitotic fiber attaches to a chromosome, the chromosome will be oriented until the kinetochores of sister chromatids face the opposite poles. Eventually, all the sister chromatids will be attached via their kinetochores to microtubules from opposing poles. Spindle microtubules that do not the engage the chromosomes are called polar microtubules. These microtubules overlap each other midway between the two poles and contribute to cell elongation, which we will see in anaphase. Astral microtubules are located near the poles, aid in spindle orientation, and are required for the regulation of mitosis. Metaphase. The change phase. All the chromosomes are aligned in a plane called the metaphase plane or the equatorial plane, roughly midway between the two poles of the cell. The sister chromatids are still tightly attached to each other by cohesive proteins. At this time, the chromosomes are maximally condensed. In anaphase, or the upward phase, the cohesive proteins degrade and the sister chromatids separate at the centromere. Each chromatid, now called a single chromosome, is pulled rapidly towards the centrosome to which its microtubule is attached. The cell becomes visibly elongated, oval-shaped, as the polar microtubules slide against each other at the metaphase plate where they overlap. Telophase or the distance phase. The chromosomes reach the opposite poles and begin to decondense, unravel, relaxing once again into a stretched out chromatin configuration. The mitotic spindles are depolymerized into tubulin monomers that will be used to assemble cytoskeleton components for each daughter cells. Nuclear envelopes form around the chromosomes and nucleosomes appear within the nuclear area. Cytokinesis. In animal cells, cytokinesis typically starts during late anaphase. A contractile ring composed of actin filaments forms just inside the plasma membrane at the former metaphase plate. The actin filaments pull the equator of a cell inward, forming a fissure. The fissure is called the cleavage furrow. The furrow deepens as the actin ring contracts, and eventually the membrane is cleaved into two. So that's how from one cell you get two.
Regulation of the cell cycle. What are cyclin CDK complexes? Cyclin CDK complexes are protein complexes formed by a regulatory cyclin subunit binding to an inactive cyclin-dependent kinase, or CDK, creating an active kinase that drives the cell cycle by phosphorylating target proteins to trigger essential events. In G1, cyclin D is responsible for increasing DNA building blocks. In G1/S, cyclin E acts as a global regulator of cell cycle entry. In S, cyclin A is responsible for initiation of the DNA replication. In M, cyclin B allows entry into mitosis. Let's talk about the cell cycle checkpoints, yeah? So there is a checkpoint here called start. Before advancing to the next phase, the G1 checks for adequate size, amount of nutrients, molecular signals like growth factors, DNA integrity. G1/S and S phase cyclin CDK complexes are activated, resulting in a phosphorylation of proteins that initiate DNA replication, centrosome duplication, and other early cell cycle events that happen in S. There is three mechanisms in which progression to S is inhibited. Number one, downregulation of CDK genes. Number two, rates of cyclin degradation is increased by anaphase promoting complex or APC through addition of ubiquitin. Number three, presence of high concentrations of CDK inhibitors in G1. There are three important genes that are related to the cell cycle regulation and are also important in the development of cancer. So this is how it works. P53 works in this phase by checking there's no DNA damage, repairing it if it finds that there is, and if it can fix it, it triggers cell death, also called apoptosis. This is important because if P53 fails, then cancer can appear. Another important one is P21 that raises as P53 increases its levels and stops the cell cycle by inhibiting CDK cycling complexes. Retinoblastoma protein, or RB, monitors cell size, binds to the transcription factor E2F when the cell grows in size. E2F is released and activates genes that produce a transition protein. So in S, when we enter the S phase to ensure G1 events stop happening, there is destruction of the G1/S cyclins, and the G1/S cyclin gene expression is reduced. Towards the end of the S phase, M cyclin gene expression is switched on and M cyclin concentration rises, leading to the accumulation of MCTK complexes during G2. However, these are phosphorylated for now, so they're inactive. If the cell fails to complete DNA replication, for example, a negative signal blocks the initiation of mitosis. This is the G2/M checkpoint, by the way. About MCDK produced in S, the abrupt removal of this phosphorylation leads to the activation of all MCDKs. These then trigger progression through the G2M checkpoint. The G2 checkpoint checks for DNA damage, DNA replication completeness. The M, also called spindle checkpoint, occurs near the end of the metaphase stage of karyokinesis. The M checkpoint is also known as the spindle checkpoint because it determines whether all the sister chromatids are correctly attached to the spindle microtubules. Because the separation of the sister chromatids during anaphase is an irreversible step, the cycle will not proceed until the kinetochores of each pair of sister chromatids are firmly anchored to at least two spindle fibers arising from the opposite poles of the cell. If the APCC gets the right signals at metaphase, it starts a chain of events that destroys cohesin, the protein that glues the protein glue that holds sister chromatids together. The APCC first adds ubiquitin tag to a protein called saccharin, sending it for recycling. Saccharin normally binds to and inactivates a protein called separase. When saccharin is sent for recycling, separase becomes active and can do its job. Separase chops up the coenzyme that holds sister chromatids together, allowing them to separate. So that separation will happen in anaphase, but the checkpoint is in metaphase. So I want to give a shout out to the sources. So the first one is the Cell Cycle Principles of Control by David O. Morgan. This is a book from 2007. It has 300 pages. It is very complete, very informative. And I had to use another source called OpenStax, which is also free. Both of these are free, but OpenStax accepts donations. Um, so OpenStax is shorter, so it allowed me to digest the bigger book better. So I use both in combination to make this video. I will leave the links below. in the description of both of these sources. Thank you very much. And finally, the credits. Camila Pimentel, which is me, and the drawings, the editing, the script, the voiceover. Charlotte, thank you for the suggestions and moral support. To my family and dogs, thank you for the moral support. I also want to thank you, the person that is currently watching this video, if you made it to this point. If it's not too much, I will also like for you to give me a like, subscribe, and comment on this video. It will really, really help me a lot. Again, many thanks.